Imagine you accidentally cut your finger. When that happens, a series of steps start to take place in your blood. We call it a coagulation cascade. The coagulation cascade is a process that happens in our blood to form a clot when we get a cut or injury. It involves a series of reactions that activate different substances in our blood. First, there are two main pathways called the intrinsic and extrinsic pathways. These pathways are like different roads that eventually meet. Let's dive into the details of the intrinsic pathway of the coagulation cascade. The first step is triggering the intrinsic pathway. The intrinsic pathway is initiated by damage to the endothelium, the inner lining of blood vessels, or exposure of blood to negatively charged surfaces. These events cause the activation of clotting factor 12, also known as the Hageman factor. Activation of factor 12. When factor 12 comes into contact with the damaged endothelium or negatively charged surfaces, it undergoes a conformational change, resulting in its activation. The activated form of factor 12 is called activated factor 12 or 12A. This activation process is known as contact activation. Activation of factor 11, activated factor 12 or 12A, now acts as an enzyme and cleaves another clotting factor called factor 11, converting it into its active form known as activated factor 11 or 11A. This cleavage reaction requires the presence of a protein cofactor called high molecular weight kininogen, HMWK, and prekallikrein. Activation of factor 9, activated factor 9 or 9A, continues the cascade by cleaving factor 9 into its activated form, which is known as activated factor 9, 9A. This cleavage reaction occurs in the presence of calcium ions, platelet phospholipids, and another cofactor called factor 8. Formation of the factor 9 of factor 8 complex, activated factor 9 or 9A, forms a complex with its cofactor, factor 8, which is activated to its active form known as activated factor 8, 8A. This complex, called the factor 9A factor 8A complex, is crucial for the subsequent activation of factor 10. Activation of factor 10, the factor 9A factor 8A complex, and the presence of calcium ions and activated platelets serves as a potent activator of factor 10. This activation takes place on the surface of activated platelets, which provide a favorable environment for enzymatic reactions to occur. Progression to the common pathway. The activation of factor 10 marks the end of the intrinsic pathway and the initiation of the common pathway in the coagulation cascade. Activated factor 10 or 10A will now proceed to participate in the final steps leading to the formation of a blood clot. The intrinsic pathway, along with the extrinsic pathway, plays a crucial role in the coagulation cascade by ensuring the formation of a stable blood clot. The intrinsic pathway responds to damage to the blood vessels themselves and is triggered by contact with negatively charged surfaces. Let's explore the details of the extrinsic pathway of the coagulation cascade. Tissue damage and exposure of tissue factor. The extrinsic pathway is initiated by tissue damage. When tissues are injured, a substance called tissue factor, TF, also known as factor 3, is released and exposed to the bloodstream. Interaction of tissue factor with clotting factor 7. Tissue factor interacts with clotting factor 7 in the presence of calcium ions. This interaction leads to the activation of factor 7 to its activated form, known as activated factor 7 or 7A. Formation of the tissue factor 7 a complex, activated factor 7 or 7A, forms a complex with tissue factor, and this complex is called the tissue factor 7 a complex. Calcium ions play a crucial role in stabilizing this complex. Activation of factor 10. The tissue factor 7 a complex is a potent activator of factor 10. Once formed, the tissue factor 7 a complex activates factor 10 by cleaving it into its activated form, known as activated factor 10 or 10A. Initiation of the coagulation cascade. The activation of factor 10 marks the initiation of the common pathway of the coagulation cascade. Activated factor 10 or 10A now participates in subsequent reactions leading to the formation of a blood clot. The extrinsic pathway is named so because it is initiated externally to the bloodstream, primarily in response to tissue damage. Tissue factor is released from damaged tissues and triggers the activation of factor 7, which then leads to the activation of factor 10. Let's explore the details of the common pathway of the coagulation cascade. Activation of factor 10. The common pathway begins with the activation of factor 10, which is the result of both the intrinsic and extrinsic pathways. Activated factor 10 or 10A is formed through the action of the factor 9A factor 8 complex from the intrinsic pathway or the tissue factor 7 complex from the extrinsic pathway. Formation of the prothrombinase complex, activated factor 10 or 10A, combines with other substances to form the prothrombinase complex. This complex includes activated factor 10 or 10A, factor 5, calcium ions, and phospholipids derived from activated platelets. Conversion of prothrombin to thrombin. The prothrombinase complex acts as an enzyme to convert prothrombin, also known as factor 2, into its active form called thrombin, 2A. Thrombin is a key enzyme in the coagulation cascade. Thrombin functions. Thrombin amplification. Thrombin serves as a positive feedback loop by activating additional clotting factors. It activates factor 5, factor 8, and factor 11, which amplifies the coagulation process and leads to the generation of more thrombin. Fibrinogen to fibrin conversion. Thrombin converts fibrinogen, factor 1, into fibrin monomers. Fibrinogen is a soluble protein in the blood, and thrombin cleaves it to produce fibrin monomers. Activation of factor 8. Thrombin also activates factor 8 or factor 8A, which plays a role in stabilizing the fibrin clot. Factor 8 strengthens the fibrin meshwork by cross-linking fibrin strands, resulting in a more stable blood clot. Fibrin clot formation. Fibrin monomers generated by thrombin polymerize and aggregate, forming long strands of insoluble fibrin. These strands form a meshwork that entangles platelets, red blood cells, and other components of the blood, leading to the formation of a stable fibrin clot. The common pathway is the final stage of the coagulation cascade, following the intrinsic and extrinsic pathways. It involves the conversion of prothrombin to thrombin and the subsequent formation of a fibrin clot. Thrombin not only converts fibrinogen to fibrin but also plays a role in amplifying the coagulation process and stabilizing the clot through the activation of other clotting factors. It's important to note that the coagulation cascade is tightly regulated to maintain a balance between clot formation and prevention of excessive clotting. Various anticoagulant mechanisms and feedback loops work in conjunction with the coagulation cascade to prevent abnormal clotting and maintain hemostasis. That was all about the coagulation cascade. Keep watching for more medical knowledge. Welcome to sqadia.com, your one-stop destination for medical online learning. Dive into our extensive catalog of in-depth animated videos, meticulously tailored for students, residents, and even practicing healthcare professionals. With over 40 medical courses available, covering topics from the fundamental anatomy to the complex endocrinology and everything in between. Explore various teaching styles. Engage with our immersive videos, purposefully designed to make learning not only incredibly enjoyable but exceptionally effective. To get started on your path to excellence, take advantage of our exclusive free trial and gain immediate access to a wealth of educational resources. sqadia.com, your journey to exceptional medical education begins here.